Welcome, everyone. Today, it's a pleasure to welcome Professor Suzanne Foitzik and her PhD student, Marah Stoldt. Both researchers are part of the Institute of Organismic and Molecular Evolution, Johannes Gutenberg University Mainz, Germany. Professor Suzanne is interested in the evolution of social behavior, for which she and her team use integrative approaches ranging from behavioral ecology over genomics to epigenetics. Her main models of study are ants, which are used to tackle questions as how parasites affect and manipulate social insects. In this occasion, Professor Suzanne and Marah Stoldt will present results on the evolution and molecular regulation of the complex behaviors of Temnothorax ants, including how parasites manipulate their behavior. Welcome.
Well, thank you for the kind introduction and the invitation to give this talk today. It's a pleasure. Yes, and I will talk today about social evolution, aging, and host-parasite interactions in Temnothorax ants, and I will be joined by Marah, who will take the middle part. So our talk is structured in 3 parts. We will start with the chapter on division of labor, and then Marah will talk about the molecular coevolution of zoonotic ants and their hosts. And finally, I will talk about a case of parasite manipulation. And we mainly work on Temnothorax ants, and these ants are actually tiny. So the entire colony fits into an acorn, as you can see here. So on average, these colonies have only like 30, 40 workers and 1 to several queens, but they show quite high diversity of different lifestyles, including a social parasitic ones. And we can easily collect literally thousands of colonies, and we can do nice experiments. They're easy to observe. They're rather slow ants, but they're great models for a lot of the questions. And we work on different Temnothorax species, so you will see different species kind of appearing throughout this talk. So, and I want to start out with a chapter on division of labor. Well, when we look at ant colonies, it often looks like a lot of highly unorganized, seemingly similar looking ants running around and a little bit crazy. But when we mark the ants, we actually notice that they are rather specialized, that each ant has a certain task which they normally perform. So they take over different role, and what looked like a lot of chaos when you to the uninformed eye, it's actually quite well organized. But it's rather a degree of self-organization. So there is no queen in the morning who tells this ant to do this and this ant to do that, but each individual has a different likelihood to take over different chores, and how this is organized and the molecular regulation of it is what we want to explore here. Actually, division of labor is one of this organizational principle which is common to many different biological systems, including human society, if we think of here the conveyor belt. So we see that in multicellular organisms, different cells take over different functions, and indeed, division of labor is part of 3 of the major evolutionary transitions, including the one from solitary to a social lifestyle. And here we look at social insects. And actually, Adam Smith already in 1776 said that societies where individuals are more specialized have a higher productivity, and this is due to a higher individual efficiency, and it also reduces the cost associated with switching a task if you always do the same task. In social insects, we find different layers of division of labor. We find the division of labor between reproductives, the queens and the workers in ants, but there is also a secondary division of labor, which we call worker specialization. And here, as I said, it's not the queen who gives the ants the different tasks, but rather it's rather self-organized. And these are the models we use to explain how this task division is actually working. So individuals have different response threshold to respond to a certain cue. So imagine you have a larvae crying out for food. So some individuals have a low threshold, so they will take care of the larvae and feed it. Others have a high threshold, and because the ones with a low threshold do it, the other ones will normally not do it. And these thresholds are influenced by gene expression, which in turn is influenced by nutrition, by age, by fertility, and also by the demands of the colony. Often we have a positive feedback loop. So if you start doing a certain task, your threshold for that task will be lowered. So you are more likely to do it again and again. Whereas in most social insects, it's assumed that age has a strong effect on division of labor. There were some studies which suggested that in Temnothorax ants that might not be so important, but rather the nutritional state is important. So we checked out this in the ant Temnothorax longispinosus, which you can see here, which is from North America. And indeed, we could show that young workers take over mostly brood care, whereas older workers go out and foraging. When we look at nestmate care, this is kind of grooming or feeding of other workers and the queen, we found that this is basically equally done by young and old individuals. So we too find the typical age division of labor, which has been shown for many social insects. The next questions we had is how flexible are workers, which genes are linked to task, and how are those genes regulated? So these are experiments which were done by a PhD student of mine, Philip Kohlmeier. And what he did do is he either took away all the young workers, so all the brood carers, or all the old workers, or half of both. And in case there were no brood carers left anymore, then foragers basically were forced to go in and change their task. And so with this experiment, he was able to disentangle age from task, and also he looked whether an individual was fertile or not. So he could show that it's much more difficult to get young workers to go outside and forage. So he didn't really, over a short time frame, get a strong increase in foraging in young workers, even though the foragers were taken away. When we did the reverse, so basically took all the brood carers away and forced foragers to go back, he actually found that this is really happening. So older workers go back inside and take over brood care. So going outside, it's more difficult than coming back inside. The next thing is he did a gene expression study, and he used these ants from this experiment, 48 workers, and he did whole body transcriptomes. And he wanted to see with this data how much that age, task, or fertility influenced gene expression. And in his study, he could show that actually behavior, that is whether an ant was a nurse, cared for brood, or was a forager, explained much more from the gene expression than age or fertility did. So it's really the behavior which was linked to most of the differential gene expression. And one gene he found to be very differentially expressed and highly upregulated in young nurses was a copy of vitellogenin. And what vitellogenin was already known in the honeybee to be upregulated in nurses and actually downregulated in foragers. And there is another, the juvenile hormone plays in this regulation also a major role. So we actually investigated the copy of vitellogenin that we found, and this was vitellogenin-like A. So vitellogenin, actually, there was a gene duplication which occurred in ants and also in some of the other hymenopterans. So there are several copies of this gene, and we already found in an earlier study that queens upregulate certain copies versus workers upregulate others. This vitellogenin-like A, we did a large-scale phylogeny to find, so where is it located? And actually, it's up here, and it is not the same copy than the one known for the honeybee, which is down here. So basically, there is a different vitellogenin gene which controls brood care behavior in this ant, whereas in the honeybee, it was a different copy of vitellogenin, which in our case was not linked to brood care behavior. So we wanted to find out, is this gene really important to control brood care behavior? And to find this out, we did an RNAi-mediated lockdown. So we did it by feeding the ants double-stranded RNA of this gene, and we had a control of nonsense RNA. And we could show that we were able to downregulate the gene in the fat body by about 70%. And it was mainly expressed in the fat body. We then observed the ants, which had this downregulation and no downregulation. And lo and behold, we can show that individuals where we knocked down that gene reduced the brood care behavior. So indeed, this gene was causally responsible for controlling brood care behavior. We did a control experiment to find out, because we treated the entire colony, so in theory it would have been possible that our treatment of larvae yielded this effect. So we did a cross-fostering experiment, but this shows that our RNAi treatment really affected only the workers and not the larvae. Okay, so we can knock this gene down and the workers stop doing brood care. So do they go out and forage? Well, we didn't find that. We didn't get an increase in foraging, but we did get an increase in nestmate care. And you can see this here. So a 6-week lockdown leads to an increase in nestmate care behavior in knockdown individuals. So they don't progress all the way to become foragers, but they do progress to kind of the middle, the nestmate care term. And this is actually interesting because brood care and nestmate care are from the behavior very similar. You antennae, you groom, you feed, you maybe carry your nestmates. Either you do it for brood or for adult workers. But who you do this behavior to changes from brood to adult workers. So basically, we get this shift that they are interested in brood, that they are interested in other nestmates. So we wonder whether the Q responsiveness really changed. So what we did is we extracted the cuticular hydrocarbons, which are the hydrocarbons basically are the signals which indicate you are a worker or you are a larvae. We did these extracts and we presented ants only with the extract. And then we looked that we did this for knockdown and non-knockdown individuals. And when we do this, we can actually show that when we knock down the genes, young workers shift their interest from mainly to look at the brood odors to the nestmate odors, suggesting that indeed this gene controls the perception of brood versus nestmate odors, which was really interesting because it basically means that its effect is over Q responsiveness. The next question is, okay, how are these genes which are differentially expressed and related to behavior regulated? And there was an earlier study by Simola and al., which showed that histone acetylation might play the role here. So we did an experiment by feeding the ants with C646, which inhibits the histone acetyl transferase. And then Philip did the same experiment again. So he forced workers from being brood carer to become forager. But now he had one system where he had this inhibition and another system where he didn't. And when he did this, he found actually that that the inhibition promoted the switch from nurses to forager. Whereas when he did the reverse, forced foragers to go back inside, this was impeded, suggesting that histone acetylation does play a role, but it's not in the same direction. And actually, it looks like a high activity of histone acetyl transferase keeps workers in this brood carrying mode. The ultimate reason could be that this high activity prevents young workers to leave the safety of the nest too early in life. As I said before, that histone acetylation was also found to play a role in Camponotus ants, but here actually it looked rather the reverse, that the high activity was linked to foraging. We are currently analyzing the ChIP-seq sample from this experiment to really pinpoint which histones were different acetylated and whether they were indeed linked to the differential expression of genes. In another set of experiment, Marcel, another PhD student in my lab, looked at gene expression in the brain, but now also in the antenna. And we included the antenna because we saw that this cue responsiveness was largely important for kind of the shift in behavior. And for the brain, he also did ChIP-seq analysis and microRNA. So not only to get the genes which are differentially expressed, but also to look at the epigenetic regulation of those genes. So what he found was that actually many more genes are differentially expressed in foragers and nurses in the antenna. So this was 25% of all genes expressed there. And here he did not control for age, so we had young nurses and old foragers. In the brain, we also found 333 genes to change the expression, but this was only 4% of all genes, suggesting that maybe worker behavior depends more on signal perception than on brain activity. And actually, when we looked at what kind of genes change the expression in the antenna between nurses, so the young brood care individuals, and foragers, we actually found that, that half of all odorant perceptors are differentially expressed. So odorant perceptors' function is to basically bind odors, and as ants mainly communicate chemically, it was interesting that over half of these odorant perceptors were differentially expressed, suggesting that if you're a forager or a nurse, really puts you in a different chemical environment, if you wish. So you probably do different things because you perceive different odors. To some these experiments, and these are the first results of our ChIP-seq analysis. So he did find indeed in the brain data that we have forager and nurse histone. We have histones which are differentially acetylated in nurses and foragers, and we identified genes close to those histones. And we actually could do a geo-enrichment and we found that ATP-dependent chromatin remodeling is one of the enrichment based on these differentially enriched or differently histone acetylated, differently acetylated histones. So. And then we were interested, okay, do we have overlap between differentially expressed genes and genes which are close to histones which are differentially acetylated? And indeed, we find some, and one of them was very interesting. And this gene is called helicase SWAP, and it's actually a co-activator of CRAB-mediated transcription, and it also fits to our enrichment of ATP chromatin remodeling, and it was higher expressed in nurses. And this is interesting because both in the Camponotus ants and also in the honeybees, this gene and crab binding protein were already found to control worker behavior. So it looks like maybe the individual genes are different ones. I told you about vitellogenin and vitellogenin-like A not being the same, but maybe the control mechanisms are largely actually similar. To sum this up, while we do find age polyism in Temnothorax ants, young workers stay inside, olders go outside. Gene expression is largely affected by tasks, so what an ant does, and much less by age and fertility. Vitellogenin, one of our candidate genes, is mainly expressed in the fat body, and it affects the responsiveness of ant workers to certain cues. And when we knock it down, then we see a behavioral progression, not all the way to forager, but to nestmate care. And it looks like histone acetylation plays a role in controlling these genes, and we are still working on this. But one thing is very interesting is that actually nurses and forager strongly differ in antennal gene expression, and a lot of their odorant receptors were differentially expressed, suggesting that they live in different, let's say, odorant worlds, and this might explain the differences in behavior there. And with this, I would like to hand over to Marah, who continues on the molecular coevolution of zoonotic ants. Thank you.
Thanks, Suzanne. So a general perception many people might have is that all the ants are hard at work. But is this really the case? And you might imagine, because I'm asking like this, no, that's not the case, because there are some ants which prefer to let others work for them. And these are called the social parasites. Parasites in general are characterized by exploiting the resources of their hosts, and these resources can be manifold. So some might exploit nutrition, while others just exploit metabolic pathways, or they can even exploit behavior, as we know from this famous cuckoo example. And very often, I mean, in all cases, this exploitation is beneficial for the parasite, but very often detrimental for the host, causing reduced fecundity or longevity. So you could say that parasites are living at the expense of others. Ankylosis is one form of social parasitism in ants, which was formerly known as slave making. And it interestingly evolved at least 10 times independently within the ants. And we will focus on this form of social parasitism in the following. So let's have a closer look at the life cycle of a zoonotic ant. So here I use the example of Temnothorax americanus, you can see here on the left, and its main host Temnothorax longispinosus here on the right, which is one of the study systems we use the most in our lab. So here we have a parasitic colony in red with some with at least one queen and some parasitic workers, as well as host workers in black. And so a young parasitic queen will go out to mate, and after she has done so, she will search for a host colony to found her colony in. So she will drive away and or kill all the adult individuals in there, and in the end, she will remain with at least some of the host brood. From this host brood, of course, host workers will emerge. And interestingly, these host workers will directly start to take care of both the parasitic queen, but also of her parasitic brood. And again, from this parasitic brood, 1 year later, parasitic workers will emerge. And so the colony will start to grow. And of course, the demands will be higher. So at some point, they will need more host workers. So during summer, the parasitic workers, which usually don't forage or care for the brood, they have basically one task. They will go on so-called raids, where they will find a new host colony to steal some nice pupae and transport it back into the parasitic colony, where from this brood, new host workers will emerge. And because the raiding as well as the colony founding step are very detrimental for the host colonies, they are under strong selection to develop respective defenses against these social parasites. On the other hand, the parasite is dependent on its host because it does not do the normal worker chores itself, so it is under selection to evolve respective counter defenses. And this reciprocal dynamic is also known as the coevolutionary arms race. So here I just quickly wanted to show you how such a raid looks when you do it in the lab. So here we have a little raiding party of T. americanus. And here downstairs, we have the host colony with some brood in white and the queen residing on the brood here. And now the T. americanus workers will try to invade this colony to be able to steal this brood. I will quickly fast forward a bit. So this is how it will look in the end. So most of the host brood is gone from the nest. It was captured by the parasites or maybe evacuated by the hosts. And you see that also all adults are basically driven out the nest, so including the host queen, which now is here. So in our lab, we focus on two questions in these systems. So the first one is, how did dolosis evolve actually? And the other one is whether and how hosts evolve defense traits against these social parasites. And for this, we mostly use ants from the Formicoxenos group. This is a really interesting group of ants because it was described as a hotspot for the evolution of dolosis, because it harbors at least 6 independent origins of dolosis. And here you can see the Temnothorax ants, which we mostly focus on. And here again, we have Temnothorax americanus I just talked about. So the first project I want to present is about the loss of perception in these dolotic ants. So from many parasitic systems, also outside of ants, we know that because parasites exploit host traits, they themselves will no longer need the genes underlying these traits, causing relaxed selection on these genes, which in the end causes gene loss. And in the case of these dolotic parasitic workers, we would expect that because they do not perform normal worker chores, such as foraging, for example, they would also not need the genes underlying these tasks. And another fact, for example, they are also fed by host workers. So everything that has to do with this search for food and maybe also food sharing might lose importance in these ants. And so Evelyn Jongepier did a project where she sequenced the genomes of eight ants, including, again, Temnothorax americanus, but in total containing 3 independent origins of dolosis. And she especially was interested in chemoreceptors. And what she found is very interesting. So here she found that in the parasites, here in red, there is a loss of up to 55% of gustatory receptors in all 3 systems. And more interestingly, even that there is also a convergent loss of odorant receptors in all 3 systems, meaning that they tend to lose the same odorant receptors, suggesting that some odorant receptors might lose importance, but odorant receptors that are important for other tasks, for example, for detecting a host nest during raiding season, might still be important and are thus not lost. So especially from Temnothorax americanus, which were often sampled from various locations in North America, we know that the ratio of parasite to host colonies varies a lot. And of course, this triggers differences in reciprocal selection on traits that are involved in host-parasite coevolution in these different populations. And I quickly want to highlight what I mean with this. So we will have some populations like the New York populations where many parasites are present. And so the selection pressure will be very strong on hosts, while there are other populations, for example, Maine, where no parasites are present or only very few. And so hosts are under less strong selection, at least for defenses. And there were several projects in our group that focused on how these differences in the parasite prevalence in host populations affected host defense traits. So, for example, one project showed that with increasing parasite prevalence in the population, hosts shift their defense strategy from fighting the parasite when they are from a host populations with no or only few parasites present to fleeing rather. So there's a shift from fight to flight. A next project also showed that colonies from parasitized populations showed less division of labor. So the workers were more generalists, which was also shown to fare better against social parasites. And also there was an increase in aggression with increased parasite prevalence in both Temnothorax longispinosus and Temnothorax curvispinosus populations. But I have to say that this increase was against conspecific non-nest mates, and this could not be found against the parasite itself. So while the other 3 projects focused on the effect of parasite prevalence in the host population, Rajbir Kaur, who also did a postdoc together with Suzanne, she also wanted to look at the parasite population. So what she did, she looked at brain gene expression and behavior during nest defense, this time using a full factorial design, incorporating both the parasite prevalence in the host population, which we from now on call parasite pressure, as well as the parasite prevalence in the parasite population, which we call parasite success, because in a population where many parasitic colonies exist, we would expect the parasites to be especially successful in invading host colonies. And so what she did, she collected Temnothorax longispinosus colonies from 8 different populations, as well as Temnothorax americanus colonies from 4 different populations. And during these nest defense trials, she noted down whether hosts attacked or not attacked the parasite. Afterwards, she extracted RNA for RNA sequencing from the host defenders and the cuticular hydrocarbons, which are important for nestmate recognition from the parasites. And so on the level of behavior, she saw that the percentage of host defenders was actually dependent on an interaction between parasite success and colony size, while the parasite state, so whether it was still freely moving around after the trial or pinned down by the host, depended only on parasite success. So meaning a successful parasite would be less likely to be pinned down by hosts, basically. And there was no effect of parasite pressure, which first came surprising. And the same is basically reflected on the level of brain gene expression. So here only a single gene depended on parasite pressure, while more than 400 genes depended on parasite success, and also more than 260 genes depended on whether a parasite was attacked or not. And these 2 sets of genes actually also showed some overlap. So genes that were highly expressed in hosts facing successful parasites were also highly expressed in hosts not attacking the parasite. So we have kind of a negative correlation that you can see here. So how does parasite success actually influence the encounter outcome? And this is where the cuticular hydrocarbons come into play. So these were analyzed with a special focus on so-called recognition cues that were previously identified. And what we found was that successful parasites carry less of these recognition cues. And this suggests that parasites are not attacked because they are able to deceive their hosts by staying under the radar. And the nice analogy is that this is similar as to wearing the cloak of invisibility. So this project focused on the population level. So how does parasite prevalence in the population influence hosts? But the next project wanted to look more on the level of the colony itself. So for this, we used a different parasite host system. We used the parasite Habroxenus sublevis here on the right and its host Diptothorax acervorum here on the left. And we performed a cross-fostering experiment. So we transferred pupae of L. acervorum either back into their natal colony, into a conspecific colony, into a heterospecific colony of a different host species, or into a parasitic colony. And then we waited until these pupae emerged into workers, let them live there for 3 months. Afterwards, we recorded their behavior and then sampled them for RNA-seq. And with this, we wanted to first answer how the adult social environment, including the parasitic nest, affects both brain and internal gene expression, but also more generally how the behavior performed 1 hour before sampling affects both brain and internal gene expression. So for the first question, we found that actually a number of genes changed in the brain with these changes in the social environment. And very interestingly, most of the genes differed between the parasitic treatment here in yellow and the other 2 treatments. So this suggests that hosts can distinguish between a parasitic and also a conspecific or the natal nest. In the antenna, the picture was a bit different. We found less differentially expressed genes, and we found a stronger difference to the conspecific treatment, so here in green. And we also identified several candidate genes. Some of them were related to lifespan, suggesting that lifespan could be altered in the hosts living within the parasitic nest, but we would need more behavioral observations and probably also long-term studies to really prove that this is the case. To the second question, so we tried to correlate the behavior 1 hour before sampling together with the gene expression by performing a WGCNA. So we constructed basically a gene network. And then we saw that brain transcriptomes did not correlate to either behavior or location, but antennal transcriptomes actually showed at least some correlations. And this kind of fits what Suzanne showed us in the first part, that in Temnothorax longispinosus, antennal transcriptomes were actually better in explaining behavioral tasks than brain transcriptomes were. And this is really interesting because we mostly work on the molecular basis of behavior, and this suggests that we should always consider to also sequence antennal transcriptomes and not only brain transcriptomes. So last but not least, I just wanted to show, we also found one very interesting module, which is not only correlated with behavior and location, but also harbors 11 differentially expressed genes, which is more than expected by chance. But this linkage between the behavioral results and this difference in the social environment, we would need more behavioral observations to really prove that host behavior is affected by living in a social parasitic nest. So to sum up, we found that parasites lose odorant and gustatory receptors due to their new lifestyle. Parasitic ants and their hosts engage in coevolutionary arms races, and host populations under strong parasite pressure are more aggressive, flee rather than fight, show a lower division of labor. Moreover, we could show that host brain gene expression and behavior depended on where the parasite is from, not where the host is from, and that parasites managed to not get attacked by going under the radar. And we also show that within the parasitic colony, gene expression of exploited hosts was affected. We just don't know yet how. And with this, I will give over to Suzanne again.
Okay, thank you. Yeah, the last story I want to tell is again a story about a parasite, also an obligate parasite as our social parasites before, but it's a real parasite if you want. Is this working? Oh yeah, okay. So parasites can manipulate host behavior and also cell life history. I mean, you probably know some of those stories, like of the Lancet liver fluke here, where parasitized ants change their behavior and climb up on grass stems and bite into it. Or of this ant here, Cephalotus atratus, where infected workers actually get an abdomen and also show deviant behaviors. There are fascinating stories, and the question is, why does the host or the parasite do that? The interesting thing is we find most often these changes in parasites which have complex life cycles. These are parasites which use different host species sequentially. So basically they have an intermediate host and they have a definite host. And in the definite host they do sexual reproduction, but they need the intermediate host basically to reach the definite host. And most of these manipulations are therefore a means by which the parasite often tries to get into the final host or the definite host. But how does it do it? I mean, the proximate mechanism is currently a hot topic, and we investigate this in the following system. Here we have the final host or the definite host, which is a woodpecker, and the intermediate host is an ant here. And the woodpecker is infected by a tapeworm, and the eggs of the tapeworm are given away with the feces of the bird, and for whatever reason, these ants are collecting this feces and are feeding it to the larvae. And once adult workers emerge, we see a different phenotype. So here to the left, a brown worker, this is a healthy worker, and the yellow worker is infected by the cestode. The cestode is here in the cystocercoid stage, in a larval stage, and you can see it here in these two pictures. It looks more like a little, egg actually, and it lives in the haemolymph, loosely attached to the gut, and the individuals can be infected by one cestode up to 70-something cestodes. So the parasite load can vary. And basically the life cycle is finished when the woodpecker comes along, opens the acorn with these infected ants, eats some of these eggs with the brood, and gets infected. We have at the moment only worked on the cestode, the ant part and the cestode part of the story. And this is work done by Sarah Beros, a former PhD student, and Inon Shah, a postdoc. And they just observed these ants, and they found that infected ants are actually slightly smaller. They stay close to the brood in the nest. They are, when we look at the behavior, they're here in yellow, infected ants. So they rest a lot, they groom themselves. They get very often fed compared to their nestmates and workers from healthy nests. And they actually don't do a lot of the work. So they rarely ever forage or carry or feed or guard or any of this. And when we open acorns with infected ants, everyone runs away except the infected ones, which really just look up and don't do anything. And I mean, obviously, this can lead to an increased transmission to the final host. One thing which Sarah noticed was that actually, as she termed it, all these yellow workers never die. So she did a 4 months first study where she could show that indeed we have a much higher survival of cestode infected ants compared to ants from non-infected nests, but also compared to their nestmates. And actually the nestmates of infected ants die at higher rates. So they pay a price of having this infected nest. So indeed on a colony level, it is really parasitism, even though the individual itself seems to live longer. So we also find changes in these workers which are not themselves infected, but they live with infected ants. For one thing, and I will show you in a second, we find changes in behavior, but also we find a decreased survival. And here to the changes in behavior, well, we find, it also has been shown before, that the cuticular hydrocarbons are different of infected ants and non-infected ants. So you can see this here in the PCA, in yellow again, the infected ants, and you see they are somewhat different from the others. We actually show when we observe behavior in the nest that infected ants, they are very much cared for, they're fed and everything, but they're also sometimes attacked. So somehow these ants notice, okay, this is something different. So we wondered, so why do they receive so much care if the others can detect that something is wrong? So we observed the nest and we noticed that in infected nests, we have simply a lower aggression. So what we did is we removed infected workers from infected nests, and then we did aggression tests, and we saw that the aggression went up. And we did also the reverse. We put infected workers into non-infected nests, and we could alter their aggression. Meaning, and this is here the aggression of non-infected workers. So basically, only the presence of infected workers in the nest altered the aggressive behavior of their nestmates. So suggesting there is a long arm of the parasite, if you wish. And this could also explain why these infected workers, despite their slightly different chemical profile, are well accepted. Well, coming back to this observation of Sarah that infected workers live so much longer. So she actually did a 3-year study and she marked newly emerged workers and also queens which were already adult at the start of the study, and also infected workers which were already of unknown age when we started the study. And then she observed what happens to these individuals over the next 3 years. So over Sarah's entire PhD in the end. And what she could show is that infected workers survive as well as the queen do, and much, much, much better than every other worker in this nest. And here again, we find the difference that workers from healthy workers from healthy nests survive better than from infected nests. But infected workers really showed a high survival. I mean, as I said, they were already adults when we started the study. And after 3 years, more than half of them were still alive. So apparently, this cestode leads to a strong lifespan extension in workers. So it's actually beneficial from the individual kind. And then the question for us was like, how is this possible? How can a cestode, a parasite, increase the lifespan? And we think it's like 3 or 4 times. And I know a colleague who told me that at least he had one infected worker over more than 7 years. So, I mean, in the species, actually, queens can get 20 years. So, I mean, it's obviously difficult to study within a PhD. Who wants to do a PhD over 7 years? So it suggests that actually the cestodes are somehow a fountain of youth. So individuals which are infected live much longer. So the question is, how is this possible? Well, we did, and as I said, the queens in the species can get 20 years. So we were wondering, are infected workers actually showing a physiology similar to a queen? So we did some analysis, and here we can see that actually the metabolic weight is not like the queens, which is very low, but rather like young workers. And also the lipid content is actually quite high, so they're quite fat, like young workers. So actually they seem to resemble young workers more than they resemble queen. They do receive a lot of care. So when we compared, in some studies, we even had more care than the queens receive, but definitely much more than young workers and old workers do. So they're super well cared for. And here we did actually some chemical extractions, and we could show that workers from infected nests are much more interested in odors of infected workers, suggesting that somehow the cuticular hydrocarbons of these workers make the other workers take more care of them. So it looks like infected workers somehow miraculously remain in this young state. They're not really resembling in many traits the queens. In many social insect, queens get older. And in honeybees, it has been shown that this is in part due to a higher resistance to oxidative stress. So we wanted to find out whether also infected workers show a higher resistance to oxidative stress. And we did this by experimentally eliciting oxidative stress by using this poison called paraquat. And what we can see here in this 10-day study, well, normal workers don't like paraquat, they die. So about half of them were dead after 10 days. We didn't get a lot of dead ones, I think none actually among the infected workers. So they seem to withstand this oxidative stress treatment very well, suggesting that in part the extension in lifespan can be explained by a higher oxidative stress resistance. There are some parasites which extend lifespan in the host, so this is not the only case. And in many cases, this is due to castrating the host. Well, workers are normally not fertile, but when we remove the queen, actually workers can get fertile. And we know already in other Temnothorax ants, when we remove the queen, workers live longer. So we were wondering, are these infected workers actually castrated or are they fertile? We found out that when the queen is there, they normally do not lay eggs, but when we remove the queens, it's the infected workers, the first ones who develop their ovary, and here you can see eggs in their ovaries and actually laying eggs. So it's clear that it is not castration which lead to the extension in worker lifespan, because, well, they are at least as fertile as other workers are. So who benefits from this entire lifespan extension? Well, we think actually the parasite does, because these infected workers are not leaving the nest and exposing themselves to the woodpeckers, but they stay in the acorn. So the woodpecker has to come along and eat the infected ants. And if they live longer, if they live decades, let's say, then the likelihood that eventually the woodpecker comes along is much increased. So here the parasite could act as a puppet player. And we did a study, in this case it was Barbara Feldmayer, who looked in my lab at the gene expression in the brain. And indeed, we found between infected and non-infected workers genes to be differentially expressed. And a lot of these genes had, again, functions in the fight against aging and oxidative stress, like transferrin or this peroxidase, suggesting that actually there is an upregulation of anti-aging genes. Another thing we found is that infected workers downregulate muscle genes. And actually, when we checked the muscles, we found that infected workers have actually a lot of gaps in their muscles, whereas healthy workers have these normally striped muscles. So this could also explain the high inactivity we actually see in these infected workers. So Marah here did a study where she compared gene expression in the gasternal and the abdomen between infected workers and the queens to see whether there is an overlap in gene expression, to again see, is a parasite here utilizing the gene networks also present in queens. However, we actually didn't find a lot of overlap. There were only 6 genes which were commonly upregulated in queens and infected workers. However, among these 6 genes. There was one, which is a well-known candidate as an anti-aging gene, carboxypeptidase B-like, which was called in Drosophila the silver gene, and it is linked to postponed senescence. So maybe, but there is not a strong overlap. And both the queens and the infected workers upregulate genes with immunity functionality. And the final bit I want to tell you is, okay, the cestode is living in the haemolymph, in the blood of the ant, if you wish. And so we were wondering whether the cestode is releasing anything into the ant. So we did, together with Juliane Falk-Butter and Alessandro Carrion-Noriega, a study where we looked using proteomics at the composition of the haemolymph. And what we found is, and here you compare the haemolymph protein from infected workers to healthy workers, and every red dot is actually a protein by the cestode. Indeed, 6% of all proteins found in the haemolymph of infected workers are cestode in origin, are parasitic, which I find a very high number. We also found proteins to be differently common from the ant, but these cestode proteins, the majority have a clear secretory signal, so they were secreted into the ant. Unfortunately, only 30% of those secreted proteins we can find in annotation. And that was actually very surprising, because when we do a proteomic analysis of the cestode itself, then we found that over 80% of the proteins are annotated. What does that mean? It probably means that proteins given into the ant are novel genes, at least genes where we don't find homologs in related species. Actually, you can see this here. So these are the top 50 proteins found in the ant from the cestode. And everything in gray, we don't know what it is. We only know it's from the cestode. And you can see that most of them have no annotation. The ones we do have actually interesting annotations. So these two here are clear antioxidants. They have been shown in other studies, including ants, that they help to fight oxidative stress, which is actually interesting, which means that this parasite is releasing antioxidants into the bloodstream of the ant. And this could explain their high oxidative stress resistance and also, in part, their high lifespan. To sum up the story, well, infection with the cestode Anomotaenia brevis induces changes in behavior, the chemical profile, morphology, and lifespan in ant workers. Infected workers live longer. They show indeed survival rates over 3 years, which matches those of the queens. Well, why? Well, we think it increases the transmission to the final host, because eventually maybe a woodpecker comes along and eats these infected workers. Infected workers remain in a physiological young stage when we look at metabolic weight and body fat. They maintain a high reproductive potential. When we put the queen away, then they actually are the ones who lay the eggs. They receive most care from their nestmates, and they themselves upregulate genes which known functionality in lifespan. But in addition, we can show that the parasite, the cestode, is releasing proteins with antioxidant properties into the ant. And a lot of these proteins, we actually don't know what they do. And we really would like to find out, because, I mean, well, they extend the lifespan by 5-fold. I mean, this is really interesting. Well, thanks for your attention, and thanks a lot of co-workers who helped us to do all these fascinating studies, and you for listening, Alex Weil for the fantastic ant pictures, and the DFG and Genevo for funding, and you for your attention. Thank you.
Thank you, both of you. It's super interesting, plenty of information. And now let me see the questions that are in the chat. Mm-hmm. Amanda says, what is the benefit or advantage of parasites losing their gustatory receptors? Sorry if you already mentioned this in the talk. She said.
Do you want to answer, or should I?
Yeah, I mean, we think it's because they don't need them anymore. This will result in relaxed selection on these genes, and that is why they are not of use anymore and lost, basically. So,
And we see this pattern in many parasites, actually, that the genomes get smaller and then function because they don't need it anymore. So if you don't use it, you lose it, kind of. It's interesting because in this zoonotic ants, they are fed by the exploited workers. So they don't have to go out to forage themselves. So maybe they don't need the ability to taste different types of food. But it means you are served and fed kind of mouth to mouth, but you don't taste it. So I don't know. Bad luck for them.
Okay, thank you very much. We have another question from Said. Are there any predator-prey interactions for those studied species beside parasites?
Well, obviously the woodpeckers eat these ants. But the Temnothorax ants are rather small, and I have seen they have to relocate their nests very often. They live in these acorns or sometimes even in grass stems, and they deteriorate. I mean, the queen can get 20 years, but no acorn on the forest floor will last so long. And when they relocate, I have seen other ants like Formica or Mimica stealing their brood. Also, birds like to eat the brood. The ants themselves, they're hiding in the leaf litter. So I didn't see a lot of predatory events there. Yeah. And I think they are pretty safe in their nest sites because they have a very tiny entrance so that nobody can get in.
Okay. I have some comments from Natasha. She says, very interesting work. She said, Professor Foitzik mentioned that ants perform different tasks because they perceive different odors, implying that the differential gene expression is at the origin of the behavioral differences. But could it be that behaviors influence the genotype? How can we disentangle the cause from the consequence when using this type of methodology?
Yes, that is a very good question. Well, the genotype is certainly not changing, but the transcriptome can change. And if you, I mean, the workers who do different tasks, they often live also in different sections of the nest. So if you are next to the brood, you are mainly exposed to brood odors. So as far as I know, that the odorant receptors are not kind of differently expressed depending on how often they bind to certain odors. But there might be certainly feedback mechanisms. So what is cause and effect is not always clear here. And I said once an individual who starts doing a certain task has a higher likelihood to do the task in future, which probably suggests that the threshold is changing, it's lowering. So, it's a good question, and it's not easy to analyze this because you have to, what is cause and effect? But what we think is, if you are only able to perceive certain cues, you are more likely to respond to those cues that you perceive. But I mean, as you have seen in the experiment where we took away the foragers, it is possible that eventually the nurses or the brood carers go out and forage. And because, I mean, obviously they get hungry, and at some point, even though they have a high threshold to go out, at some point, the one who has the lowest from the remaining workers will do so. And by doing so, exposing itself to a different chemical environment outside, and that may in turn change its gene expression in the antenna. So I would agree in part that it's difficult to see what is cause and effect. And it's probably an interaction.
Thank you. Another from Natasha. She says, also, soon to be, Dr. Stoldt mentioned that parasites were less attacked by their host because they bear less recognition cues, which seems to be in line with Guerrieri in 2009. Do we know examples in which the opposite is found? Example that ants can detect the absence of a recognition cue?
I don't think so. The absence is difficult. I mean, what we could show is, okay, the host is using mainly methylated alkanes as recognition. So when we do a aggression experiment and we look what explained the question between two workers, it's mainly differences in methylated alkane versus normal alkanes or alkenes doesn't seem to play a role. And what we found now in this dulotic ants, that they carry especially, and we found it actually for several species of dulotic ants, that they carry especially less of these methylated ones, which are used in the host for recognition. They still carry some, and we still find a rather high aggression of the host towards the dulotic ants. So it's not a yes or no, but it's rather if you carry a lot, you kind of stick out a lot and you are more often attacked. So these parasites, they use actually various tactics. They also have a gland, the Dufour gland, and when they go in a nest, they smear workers with this, and then the workers attack each other. So this has been described as a propaganda substance. It's not like you get attacked or you get recognized or not, but it's often, if ants are not quite sure, is this an enemy or not, they sometimes do like less aggression, but if they are sure, okay, you stick out, then they're kind of full force aggressive. And this is what we find here. But the absence is difficult, yes.
Thank you. We have one more question from Evie George. Very interesting talk, he said. I have a question on the first part. Did the vitellogenin knockdown individuals show a shift in their movement away from the nest center?
We didn't check. I don't know. No, I don't think we did. The only thing we saw was we did an experiment where we had an arena which was half dark and half in light. And we found that individuals where we knocked down the genes were more often in the light area than in the dark area. But the ones where we didn't knock down the gene, they were mainly in the dark area. We somehow suggest that they might be more willing to move outside, even so we didn't see an increase in foraging. So they didn't went all the way. So I don't think that this one gene is controlling the entire division of labor. It's just controlling, I think, this kind of more of the first part, like the nursing behavior. But there must be other genes which are important when it comes to the foraging. Yes.
Thank you. Natasha says, thank you very much for your answers. I would like to ask a question about the part that Marah presented. She said that there is a convergent loss of odorant receptors in 3 groups of dulotic ants. She said that more than 400 genes depend on parasite success. But I couldn't understand well how you determine these parasite success. I mean, these rely on which kind. Will you expand a bit, please?
Yes, so parasite success we actually measured as the parasite prevalence, so the ratio of host to parasite colonies within the respective population. So this was recorded over, several years. So we had some data on this. And so this was basically then used for the colonies that were sampled from the different populations as the measurement.
Okay. So, for example, yes, sorry.
Maria, can I? Okay. So what we did is we did a cross-fostering experiment. So slave makers came from 4 dulotic ants came from 4 populations, and the hosts came from 8 populations. And so these dulotic ants were allowed to enter host nests, and then we waited for like an hour, and then we took host workers and looked at their gene expression in the brain. And what we found is that when we later on analyze the data, does gene expression depend on where a host worker is from, like its source population, or does it depend on where basically the dulotic ant is from? We found that it basically was explained by where the dulotic ant is from. And basically not only where it's from, but how common the parasite is in these populations. And so that's what we meant with these 400 genes are basically differentially expressed depending on how common parasites are in the source population.
Okay, thank you very much. And I was wondering, because you mentioned plenty of, I imagine you use different populations, different species. And I wonder, did you work with populations recently collected from the field, or did you work with populations that were already established in the lab for several generations or something like
that? No, these were freshly sampled.
Yeah, yes.
Yes.
We can keep the ants for several years in the lab, but they don't mate. So we cannot have new colonies founded. And these ants are super common in the field, at least the ones in North America. The European ones are a little rare, but the North American ones, basically on a single day, we can, I don't know, collect maybe 100 host colonies without a problem. I mean, they live in acorns and the density is super high. So sometimes we have in a square meter, we can have 8 colonies. And the ratio of social parasite to host colonies varies between population, between 1 in 5 to 1 in 20 or so. But they're actually super common also. So we went last year for a coevolution genomic project again. And actually even the non-parasitized population, when we search well enough, we always find some social parasites. They're just everywhere. And these populations are luckily also super stable. So we don't see a decline. I've been working on these ants for now 25 years, and I didn't see any decline in populations. They live in forests and they seem to do well. At least they seem to do well. Yes.
Oh, it's amazing. Thank you very much. And my last questions. When you mentioned the part about parasite infected with the cestode, I wonder, I don't know if you've already checked for this, how different were the cuticular hydrocarbons between the parasited ants at the level of compounds? I mean, if they presented totally different compounds or if they varied only in their proportions or something?
It was largely in the proportion. So, I mean, actually, we did several studies, and in one study, we found that foragers differ more in chemical profile from the nurses than infected workers differ. And actually, infected workers, again, look more like young workers. However, obviously, there is something in the chemical profile, because when we do these extractions and we offer it to the ants, they respond much more to this infected worker chemical profiles. We don't know exactly what it is, but it's largely different proportions. So I'm not sure whether it's really just one substance or whether it's a shift in proportions. So this we don't know yet, really. But it's not so major as you might say.
Okay. Thank you very much. We have another question from Charlotte Rocher. Just wondering, can you tell the nurses and foragers apart of you, or you categorize them based on what they are doing in the nests? Thanks for the interesting talk.
Well, foragers and nurses look the same. I mean, there's no morphological difference, and we have to individually mark them and then do observations. But then when you do observations over several days or a week or whatever, it's pretty clear. So some individuals are really doing a nursing behavior like 90% of their time. Foragers normally are not like 100% of their time outside, but they are like 30-40% outside. So it's just a kind of what we do. And what we normally assume, these workers can get 1 to 2 years old. I mean, you saw the survival rates, and the first months they stay inside, do food care, and then as they become older, they basically do go outside. But it morphologically, there is no difference that we can see. It seems to be normally age dependent, but there might be also individual differences in that some workers, maybe they have from the start other chemical cues or whatever, are able to perceive other cues. And so it's not like every worker is behaving the same. They are not there are also no clones. They are sisters, but not clones. So there might be also genetic factors which are slightly different, or developmental factors which differ, and makes you more prone to do one thing than the other thing.
Okay. Thank you very much for this amazing talk. It's super interesting. Congratulations to you and your team.
Thank you. That was kind. Hey. So
I think, no, I'm here. So now are we out of? Okay.
Yeah. Okay. Well, thank you. Okay, then. Thanks. Thank you.
Okay. So I'm sorry. So thank you, everyone. I think that's all for today. Thank you to you for watching this.
Yeah. Thank you for your attention, and bye-bye.
Thanks.
And
okay, good.